All right, well, here's our lecture for Wednesday. Sorry for the delay in getting this up. But yeah, as we were talking about in class yesterday, the this chapter in many ways is the opposite of what Monday is. It's basically exploring, given all of the evidence that we have, is macroevolution the most logical conclusion based on the evidence? And so basically it addresses this question: are the scars or the morphological evidence we see in living forms left over from macroevolution, from this universal common descent? So it starts off with basically exploring this question: how should we examine evidence from the natural world? So last chapter we talked about some maybe insurmountable challenges to the idea of macroevolution, but if it did happen, you would expect to see evidence of it, and that evidence would look like what the chapter refers to again as scars in these organisms. So, yeah, how should we examine evidence from the natural world? Well, first of all, in this chapter, they make an argument that we need to evaluate each line of evidence as a separate piece of evidence, not as a collective, as you try to figure out whether the evidence suggests a macroevolutionary view of origins or an interventionist view of origins. And so the way we do this is we ask questions like these: does it support one theory over the other? Or does it support one theory to the exclusion of the other? So as you're asking this first question, does it support one theory over the other? We'd also have to be honest that maybe sometimes it doesn't, and it actually supports both theories. It can't exclude one of them as an explanation or as a potential explanation for what you see. The other one: does it support one theory to the exclusion of the other? If the answer to this question is yes, then it would seem as though this evidence is overwhelming in favor of one theory over the other. And so even if every piece of evidence but one were to support macroevolution, you would still need to be honest about that one piece of contradictory evidence. You couldn't say something like, well, since, evidences 1 through 9 support macroevolution, therefore this evidence 10 must also support macroevolution. No, what you would say is evidences 1 through 9 support macroevolution. Evidence 10 is seemingly contradictory. And so you also have to do this for interventionist views. Even if evidence 1 through 9 support an interventionist view of origins, you would still need to be honest that evidence 10 is contradictory. And so as we look through this, we're going to start talking about some evidences in living forms and in the fossil record that may provide evidence in favor of macroevolution. First evidence we're going to talk about, or as this chapter refers to, the first scar of macroevolution. So if macroevolution were true, again, you would expect to see evidence of this in living forms in the way of scars left in these living forms. And so the first piece of evidence is what we refer to as homologies. And so there are two different ways to define a homology. The first one is structural, and so you would you would define a homology as a feature, or in this case a limb, that may be used for different functions, but develops in a similar way or develops from the same underlying structures. And so here you have human hand, the flipper of a seal, the wing of a bat, and the leg of a dog, that are all different functionally, are used for different different things in these organisms, but all develop basically the same way from the same bones and in the same pattern during development. And so this again is something that you would expect if these organisms all came from the same ancestor. If they're going to have a different life history and they're going to live in a different way, they're still subject to develop that from whatever structure they already have in place. They're limited by how we can do this. And so this would be opposed to an analogy. Analogy, a functional definition of analogy, is a structure used for the same function, but is structurally and developmentally unique. And so here are 4 different types of wings: insect wings, which have no skeletal component at all; flying reptile like a pterosaur, which is basically supported by one elongated finger; the wing of a bird, which is supported by the entire arm; and then a wing of a bat, which is supported primarily by elongated fingers. And so, an analogy, again, these are structures that carry out the same function, but have underlying structural differences. And so an evolutionary definition of these two terms, you would define a homology as a structure that comes from common ancestry. It's a shared structure in forms because of their shared ancestry, whereas an analogy is a structure that carries out the same function, but because of separate ancestry is not structurally or developmentally the same. And so this is exactly what you would expect to find if mammals all do root back to a single ancestor. You would expect to find that whatever they use their limbs for, those limbs would have had to develop from a very a similar plan, and something that would be shared among all of these groups. Some other scars of macroevolution. We see this homology in molecular features as well. So this chart is illustrating the number of amino acid differences in cytochrome C. We mentioned cytochrome C before. It has a protein. It's about 100 amino acids long, and it's something that you find in virtually all living forms. And so when you look at this, you can see the number of amino acid differences. And what's interesting is that the number of amino acid differences vary from, when you compare humans with a monkey versus if you compare humans with a rabbit, or if you compare humans with a tuna fish or humans with yeast. The number of amino acid differences varies, and it varies in a fairly predictable way. Now, it's not entirely smooth, but organisms that are more morphologically similar to one another are more molecularly similar at this in the amino acid sequence for this protein. And so here's a phylogeny built from these data. And so the numbers are representing amino acid differences, and so we can build a history based on how similar the primary structure of cytochrome C is, based on the similarities, in the primary structure of cytochrome C. And so again, it's pretty much what you would expect if you were just to compare the morphological similarities among these groups. And so again, this is pretty much what you would expect to be left over if all of these living forms did root back to a single ancestor. You would expect that as the amount of time since the common ancestor of two forms increases, the number of molecular differences in shared proteins would increase as well. So here is a similar phylogeny. This one is not rooted, and so that's why there are some differences, and this one is just for mammals. So, and it's just for mammal, it's actually just for placental mammals. And what these numbers represent is the number of nucleotide differences to account for the amino acid differences in cytochrome C. And so as you look through this phylogeny, you'll see again these mammals clustering in the ways that you would expect them to cluster if you just looked at the morphological data, or if you just looked at the amino acid differences between these groups for a single protein. And so whether you use morphological features, or you use the primary structure of a protein, or you use nucleotide differences, you get very similar phylogenies. And this is exactly what you would expect if these organisms all share a single ancestor. So again, we're starting to see what you might consider to be scars of this macroevolution. Of course, in the previous chapter, we cast doubt on whether it's even possible for macroevolution to happen. But assuming it is, what would you expect to find? And a lot of what you would expect to find, we're actually seeing in living forms. Now let's go and talk about the development of living forms. And so, again, I think I've mentioned already to you that a lot of the drawings that Ernst Haeckel did in the early 20th century in support of a biological evolution were found to be fraudulent. But some of the things that you do find to be true. So he proposed the idea that ontogeny, or the development of an organism, recapitulates the phylogeny or illustrates its evolutionary history. And so he would suggest that as a human develops, it basically goes through a sequence of its evolutionary history. So very, very early on in development, it's going to look like, an early vertebrate, and then later in development it's going to look like a more sophisticated fish, and then later in development it's going to look like a reptile, and then finally it'll start to look like an early mammal, and then it'll differentiate and it'll start to be clearly primate, and then finally it's going to be clearly human. And a lot of these drawings again were fraudulent, but what you do see is that there are particular developmental patterns that are present in all of these groups. So at various stages of development, these organisms actually look very similar, and this is especially true in the early middle stages of development, that there are certain hallmarks that you find in all of these groups that in some ways are an illustration of the evolution that may or may not have taken place. Some additional developmental scars. This is a really interesting one provided in the text. So again, you have to understand that the idea is that birds actually sit within reptilia. There's a lot of argument that class Reptilia is no longer a valuable taxon because it's what we would call paraphyletic. That is, some of the ancestors of that taxon are no longer in that taxon. And so the way we deal with that is that if you take birds and you put it inside of that taxon, now it makes that group monophyletic, including the ancestor and all of the descendants of that ancestor. What you find is when you look at the hind limbs of a reptile, you see a very robust tibia and a very robust fibula. But when you look at birds, in both the adults and the chicks, the fibula is physically attached to the tibia at the proximal end. So here up at the top, where it articulates with the femur, physically attached. And when it's attached, it does not develop. It arrests the development of the fibula. And so, again, with an understanding that birds evolved from reptiles, you would predict that they have the capabilities of developing their leg bones similar to reptiles. And so people have done experiments to experimentally separate the two, the tibia and the fibula, and what you get is the fibula developing all the way down and then actually forming ankle bones that you lack in these bird limbs. And so very interesting here, showing that birds actually have the capability of developing hind limbs that look very reptilian, again lending support that these two groups have shared ancestry, that providing maybe an additional scar for leftover from macroevolution. Then the book focuses on vestigial traits, and so it has a section just on human vestigial traits, which are right up here, but then also included in here hind limb bone in whales. And so this book defines a vestigial trait as anything that either has very minimal function or no function at all, and is best explained by having an ancestor where that was a valuable trait, and natural selection has yet to eliminate it from this group. And so the nictitated membrane in human eyes no longer needed to cover that up, to protect ourselves while feeding or while underwater. Wisdom teeth, our mouths are rarely large enough to allow these teeth to even erupt. Muscles of the nose and ears that in other animals are valuable for turning them to receive these stimuli, but in humans are significantly reduced. Body hair, again, we're not as hairy, well, most people not as hairy as other mammals, but we still have places where body hair accumulates for no reason. Some of these segmented muscles, one of the only remnants of segmentation that you find in most vertebrate groups, although found in other segmented forms. The appendix, which oftentimes even gets inflamed and has to be surgically removed, and then caudal vertebrae, which the idea there is leftover from a tail that our ancestor would have had. This one is more intriguing, the hind limb bone of a whale, but we'll talk more about that in a moment. And so these are some of the scars you would predict to find in living forms, is that they'd have some remnant of what their ancestors used that natural selection has yet to weed out. And so how you would see this play out in development would be that if you have a group of organisms that needs something for its particular life history, it's inhibited from developing something maybe in the way another group has it. It can only develop it from what it actually has. And so an example provided for that is the panda's thumb. And so panda bears strip bamboo down. They do this partly by holding the bamboo in place using a thumb that's not really a thumb. It's a thumb that develops from their wrist bones because they actually, are limited by what they can develop based on what they actually have from their shared ancestry. And so they have a thumb that's not really a thumb. It's not an opposable thumb like ours. However, it functions that way, and it's actually developed from a wrist bone. And so this is really interesting in that you have an organism that uses a particular feature, and so you could argue that it would have needed to develop that feature. But if God created that, why wouldn't he create it like the thumbs that we find in other groups? But if it actually developed or evolved, then you would expect that it would have evolve from what was already in place. And in this case, it seemingly has evolved from wrist bones. And so some other limits to what you can develop. The vertebrate eye is oftentimes used as an argument for maladaptive features, but which is what you would expect if you had to evolve these features versus if they were created. You would expect that there would be evidence of or many of these features would not be perfectly designed, but would rather just be pieced. It would look like they were pieced together with parts that were already in place as the environment basically applied selective pressure. And so the vertebrate eye is oftentimes used as an example for this. It seems that it is a ineffective design, and that the cells at the back of the eye making up the retina are actually oriented backwards, and so the light receiving portions are deep in that tissue, and light has to move through several layers of cells in order for it to be absorbed and then transmitted to the brain. This seems to be an inefficient design because it reduces the amount of light that actually gets absorbed. Another example of this is the laryngeal nerve. In fishes, comes right out from the in fishes comes right out from the brain stem, the spinal cord, and then goes straight to its destination. But in in mammals especially, and in other groups of vertebrates that have a more complicated developmental pattern, the nerve actually branches out, goes down into the chest, around the aorta, and then comes back to the larynx. And so, which seems to be a very inefficient design, and is only really explained based on the shared ancestry that we are, because we share ancestry with fishes. We have this nerve like they do, but because of how we've developed, the nerve actually has to take a very circuitous route as it moves. So another apparent scar is this hierarchical nature of life. And so what you would expect to find, maybe what you would expect to find if just all life forms were created by God, even if there were some variation within kinds, the only hierarchical nature, so here's an additional example to this hierarchical nature, that what you would expect to find is all these separate kinds should just be separate entities, and then there should be diversity within those. So it should be more like what you find in minerals. The textbook talks about this, that you have basically this whole suite of different types of minerals and crystals and other types of rocks, but there isn't really a hierarchical nature to them. Whereas in animals, what we find is that we have all these different, what we would propose are separate created kinds in cats and bears and dogs and whales, and yet we can take some of these and we can group them together based on shared features. Especially true for rodents and for primates and bats, where we have what seem to be several different created kinds, but we can group them together because of shared features. This isn't exactly what you would expect or what you would predict from a interventionist perspective, where God created these separate kinds that then diversified. You would expect to find that hierarchical nature just within the kinds, but not to where we can group several kinds into one group and several kinds into a separate group, and then maybe group those two groups together based on various features. But this is exactly what you would predict if we can root all living forms down to a single ancestor, this hierarchical nature of the organization. Biogeography is another what you might consider scar of macroevolution. And so what you'll find is that organisms, their distribution, very strongly supports in many cases, a macroevolutionary view. So one of the strongest arguments Darwin made in favor of biological evolution was a biogeographical one, that similar species are grouped together, that they have similar distributions, and those distributions are best explained by evolution within those groups. Now, what's interesting is oftentimes you have that even at the macroevolutionary level. And so when you go and you look at these groups, let's say North America. So in North America, you have these different groups of mammals, and they all are placental. And so you have placental mammals that all go and they fit these different ecological roles. They fill these different ecological niches, but they're all placental mammals. And then you look at, say, Australia, and you have basically an animal for all of those ecological roles, all of those ecological niches, but they're all marsupial mammals. And so, in addition to having the biogeography supporting biological evolution at the microevolution and speciation level, in some cases it even seems to support it at the macroevolutionary level. And then, got a couple more. Here, the fossil record. What you would expect to find in the fossil record, if all life roots back to a single ancestor, is as you progress through the fossil record, forms should become more sophisticated, more developmentally complex, and even more varied. And that's what you find in a number of these groups. The more simple forms show up early in the fossil record. As you move late within that same group, let's say algae or flowering plants, they become more structurally complex, and the variation within that group actually increases as you move through the fossil record. And so that's what you would predict to find if all these forms root back to a single ancestor, and that is in many cases what you find. Darwin predicted that as you would continue to study the fossil record, you would find enormous evidence of transitional forms. And, we don't have every single lineage modeled with these transitional forms, but we have found some groups that look to be convincing transitional forms. So here's one that's oftentimes used to support a macroevolutionary view, the mammal-like reptiles. So here we have a therapsid reptile, and then we get into a cynodont, and then finally into a living mammal. And what you can trace is what seems to be a very convincing movement of bones in the lower jaw that you find in living reptiles actually moving and becoming the bones in the middle ear, giving a good example of what we would call a homology. Features that have different functional use, but develop very similar, develop the same, because of this shared ancestry. This seems to be a fairly convincing transitional series. Another one is in whales. So we have Pachycetus, Rhodocetus, Archacetes, and then cetaceans, modern whales. And what we seem to have in these, notice these epochs here, these are all in the Cenozoic era. And so we have what are post-flood deposits. These are all flood deposits. So we can explain that one way, but these are all post-flood deposits. But we have in the exact right place, at the exact right time, a very convincing transitional series from a terrestrial mammal to a modern whale. A lineage becoming increasingly more aquatic, so therefore increasingly less terrestrial, and losing hind limbs and then modifying front limbs and modifying skeletal features in the skull, And we'll talk more about this actually in class on Wednesday. But we have, again, what seems to be a very convincing transitional series from a terrestrial form into a fully aquatic modern whale. And so, as you go through and you read this chapter, and as you listen to this, again, this chapter aims to be something basically entirely different from Monday, and it takes the perspective that, assuming macroevolution is true, what would you expect to find in the way of scars and living forms? And then realizing that we find a lot of those scars that we would expect to find. And so on Monday, what we'll do is we'll go through a chapter in the book, and the very next chapter they go through and provide an alternative explanation for each of these scars.